Hello students, welcome to EPG Pathsala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Today we are going to discuss about a module on stoichiometry of glycolysis under paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. We are going to understand the function of a central metabolic pathway that produce NADH and ATP by converting glucose to pyruvate. We will also try to understand the yield of total energy generated by these chemical reactions, and we will try to see the energy difference under aerobic as well as anaerobic condition. Living cells are self-regulating chemical engines timed to operate on the principle of maximum economy. It has been quoted by Albert Leininger. He quoted the way a living cell operates. Stoichiometry of glycolysis take place in two positions, either in the presence of oxygen or in absence of oxygen. Stoichiometry of glycolysis under aerobic condition take electron away from NADH and transfer them into oxygen to get adenosine triphosphate out. Add these two reactions up, get glucose and one oxygen, get to pyruvate 2 proton, 7 if mallet shuttle is used. There are variation in ATP yield. Under anaerobic condition, no oxygen involved. So during this step 1 to 10 are the same anaerobically electron transferred to pyruvate to make lactate, 1 glucose converted to 2 lactates and 2 proton. Make lots of lactate, then also make lots of proton, where much of the acid comes from the dissolved enamel. Most ATP yield under anaerobic conditions, but when there is no choice, then 2 is better than 0. Glycolysis is the oldest pathway and all organisms use it. It divides into two halves, start with glucose and make 2 to 3 carbon substances. Half of the pathways is getting to this point. GAP, glyceraldehyde, second half involve this glyceraldehyde phosphate being converted to product depending on aerobic and anaerobic condition. Glycolysis traditionally is represented as starting with one molecule of glucose, that is C6H12O6, and ending with two molecules of pyruvate. In addition, a total 4 ATP are generated. Through this presents a net gain of only 2 ATP. Since 2 ATP are employed to prime the pathway, that is, are used prior the generation of a gross total of 4 ATPs. The sugar splitting step is not explicitly shown but comes just prior to the conversion of NAD plus to NADH and also prior to the gain of 4 ATP. In this pathway, water molecules also evolved. So, looking at the reaction, you can see that C6H12O6 plus 2 ATP plus 2 ADP plus 2 inorganic phosphate plus 2 NADH, and these resultant product are 2 C3H2O3 plus 2 4 ATP plus 2 ADP and 1 NADH. The enzymes involved in the glycolysis pathway are hexokinase, that is first ATP priming step, phosphoglucoisomerase, phosphofructokinase, that is second ATP priming step, aldolase, that is sugar splitting step, triose phosphate isomerase, glyceraldehyde phosphate dehydrogenase, that is NADH to NAD plus 2 NADH step, phosphoglycerase kinase, generation of 2 ATP per starting glucose, then phosphoglycerate mutase, then enolase and pyruvate kinase, that during this generation of 2 ATP per starting glucose. If you see the concept map, the 6 carbon molecule glucose that began the cycle is regenerated after the 10th step of glycolysis. The 10 steps of the glycolysis are a series of redox dehydration, hydration, phosphorylation, dephosphorylation, and decarboxylation reactions. In whole cycle, 2 NADH and 4 ATP molecules are yields. If you see the stoichiometry of glycolysis under aerobic condition, glucose is converted into pyruvate and 2 molecules of NADH are formed. In the cell, NAD present as catalytic amounts, so generation of NAD plus is necessary. NADH, which present in the cytoplasm, it cannot enter in the mitochondria, so we require electron shuttles. In electron shuttle, electron pair carries NADH to mitochondrial electron transport chain and release energy in the form of ATP. In second step, malate aspartate, which mainly take place in heart and liver cell, in it electron paired eventually transferred to mitochondrial matrix, so we get 2.5 ATP per electron pair. Third is GOP-DHAP shuttle. It is a mainly step that take place in brain and skeleton muscle cells. In it, regeneration of FADH occurs, and at last we get 1.5 ATP per electron pair. This is a part of mitochondrial membrane. So electron transferred to coenzyme Q, where they join to the electron transport chain. 1.5 ATP per electron pair produced. This is the entry point which complex to skip. The first complex enters at lower level, so one of the ATP forming step is skipped. Consequences is less ATP per pair of electron. More or less ATP production depending on which shuttle is used, and also depends on cells. The stoichiometry is the calculation of relative quantities of reactant and product in a chemical reaction. Stoichiometry is founded on the law of conservation of mass, where the total mass of the reactant equals the total mass of the product, leading to the insight that the relations among quantities of reactant and product typically form a ratio of positive inters. Glycolytic stoichiometry of 2 adenosine triphosphate per glucose is the result of an optimization that maximizes the rate of adenosine triphosphate production. It is important to understand the function of a central metabolic pathway that produces NADH and adenosine triphosphate during conservation of glucose during conversion of glucose to pyruvate. Also, to examine the yield of total energy by chemical reaction and to study the energy difference under aerobic as well as anaerobic conditions. We know that during glycolysis, the 6 carbon glucose is broken down into 2 moles of 3 carbon pyruvate via 10 enzyme catalyzed sequential reactions. These reactions are grouped under 2 categories, 2 phases, phase 1 and phase 2. Phase 1, or this phase is also known as preparatory phase. It consists of 5 steps. In these reactions, glucose is enzymatically phosphorylated by ATP, first at carbon number 6 and later at carbon number 1, to yield a product that is fructose 1,6 diphosphate. It is also known as fructose 1,6 bisphosphate, which is then split in half to yield 2 molecules of 3 carbon containing compound, that is glyceraldehyde 3 phosphate. The first phase of glycolysis thus results in the cleavage of the hexose chain. It is important to distinguish between chemical reactions, which involve the balance of element and charges of all ionic species, and the biochemical reaction. For the latter, the component of the equations, that is pyrophosphate, adenosine triphosphate, etc., represent the summated ionic species for each component, and the reactant proton is not represented in equation as it is not conserved at pH 7. This approach has been followed in this analysis of stoichiometry, and it helped to focus attention of oxygen balance. We will discuss about stoichiometry of glycolysis now. In short, we can now construct a balance sheet for glycolysis to account for the fate of the carbon skeleton of glucose, the input of pyrophosphate and adenosine diphosphate, and the output of adenosine triphosphate, and the pathway of electrons in the oxidation reduction reactions. This table explains the stoichiometry of glycolysis and ATP formation under aerobic conditions. 4 ATP forms during the formation of 3 phosphoglycerate pyruvate from 1 glucose molecule. From that, 2 ATP used during the formation of glucose 6 phosphate and fructose 1,6 phosphate. The total gain of ATP is 2. So, during this step 1 and step 3, an ATP consumption occurs. During step 7 and step 10, the gain of ATP occurs. Stoichiometry of glycolysis under anaerobic condition. Under anaerobic condition, reduction of pyruvate occurs via electron shuttle. It occurs where either scarcity of mitochondria or absence of mitochondria. In this limiting amount of oxygen supply, when ATP demand is higher than glycolysis rate, also high with compared to shuttle rate. During the process, electron pair is transferred to pyruvate. As a result, glycolysis can occur without oxidation, such as any anaerobic process. Table shows total energy yield or used in every step of glycolysis. If you see in table, it is clear that during the step glucose, when converted into glucose 6-phosphate, minus 16.7 kilojoule per mole energy is generated. The glucose 6-phosphate, when converted into glucose 6-phosphate, the reaction is reversible, and again you can see the reaction is here reaction is exergonic. Likewise, it shows the energetics of each and every reactions. Overall equations and stoichiometry. Keeping in mind that each molecule of glucose yield 2 molecules of glyceraldehyde 3-phosphate, the total input and the output of all 10 glycolytic reactions is written in the first equation. Mainly three things happens simultaneously in glycolysis. Glucose is oxidized to pyruvate, NAD plus is reduced to NADH, and ADP is phosphorylated to form ATP. There can be no EMP pathway without all three events, which means that NAD plus ADP and inorganic phosphate as well as glucose must be present. Total energy yield in glycolysis, where inorganic phosphate is orthophosphate, in which the negative charge is delocalized to oxygen atom. NAD plus is nicotinamide adenine dinucleotide and can be reduced to NADH by accepting two electrons and a proton. Reactions to gain NADH can be called a hybrid transfer. It is hydride transfer, but it must be kept in mind that electrons of a breaking bond, that is highest occupied molecular orbit, attack NADH, which has lowest unoccupied molecular orbit, and no pure hydrogen occurs in reaction. In the first reaction, enzyme hexokinase phosphorylates glucose and 1 ATP is consumed. This reaction is notable for three reasons. Hexokinase is one of the regulated enzymes in the pathway. Phosphorylation traps glucose in the cell because of two negative charges in glucose phosphate. Phosphorylation destabilizes glucose, making way for further catalysis. Third reaction in glycolysis is also a phosphorylation reaction. In this reaction, the enzyme phosphofructokinase catalyzes the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. Phosphofructokinase is the key regulation enzyme in glycolysis and sets the pace of the reaction pathway. At equilibrium, 96% of the triose phosphate is dihydroxyacetone phosphate, but only glyceraldehyde 3-phosphate can react further in the glycolytic pathway and consume. The equilibrium is never reached and no DAP is left over. The sixth reaction belongs to the payoff phase. By enzyme glyceraldehyde 3-phosphate dehydrogenase is converted to 1,3-bisphosphoglycerate. Actually, six reactions consist of 4 steps. In literature, sometimes three reactions are shown because reduction of NAD plus is not always counted and has thioester intermediate. However, the important issue to understand is that this reaction produces a molecule with high phosphoryl transfer potential, that is, a molecule that can phosphorylate ADP to ATP is formed. It should also be noticed that the reaction consists of an oxidizing step where the reaction intermediate is oxidized and NAD plus is reduced to NADH. To maintain the redox equilibrium in a cell, the NADH formed must be oxidized later. As may be easily guessed, in the seventh reaction, 1,3-bisphosphoglycerate, 1 ADP to 1 ATP. The last reaction to mention 10th is also the last reaction of glycolysis. And another molecule of high phosphorylation potential, phosphoenol pyruvate, is converted to pyruvate and ATP is formed from ADP. Pyruvate kinase catalyzes this reaction and is the last enzyme in the reaction pathway subjected to allosteric control. To represent the stoichiometry of glycolysis, we require to value the thermodynamic equilibrium constant. Looking at the table, the steps that I just discussed have been shown in this flow chart and table with related enzymes and thermodynamic equilibrium constant. Also shows the stoichiometry of glycolysis. So students, let us summarize what we have learned in this module. In the overall glycolytic process, one molecule of glucose is converted into two molecules of pyruvate during the pathway of carbon. Two molecules of adenosine diphosphate and two of pyrophosphate are converted to two molecules of adenosine triphosphate, the pathway of phosphoryl groups, and 4 electrons as well as 2 hydride ions are transferred from two molecules of glyceraldehyde 3-phosphate into two molecules of NAD plus, the pathway of electron. So students, this is all about stoichiometry of glycolysis. Thank you.